I'm really excited to be here and share with you the work that's ongoing in my lab. So we're interested in trying to understand both the genetic and the molecular mechanisms of cognition. And so we're trying to ask what features of cognition are unique to humans, and we're doing this by taking a comparative approach, which I'll outline to you in this talk. In addition, we're interested in how these features might be related to the emergence of what we think of as human-specific disorders, such as autism and schizophrenia and other cognitive disorders. So we're using two main approaches, as I already mentioned, comparative genomics to identify these human-specific elements, followed by some functional analysis, and I'll give you one example of that today. In addition, we're using multiple measurements of human brain activity to identify what we think might be genes that are relevant to human cognition. So here's, again, just an outline of our overall approach. We're taking this comparative genomics approach. We're also doing this measurements of human brain activity, which I'll describe in more detail in the talk. And through both of these approaches, we think that we're able to identify genes that might be important for human cognition. We can then take the genes that are identified and do functional studies in human neurons. In addition, we're using a number of model systems, primarily mouse models for now, to do functional studies. But what's really exciting about both of these approaches is that if we do transcriptomics in either of these, we can then now identify additional genes. And so this is kind of a reiterative process to identify more and more genes that we think might be important for cognition. So here's just an outline of what I'll talk about today. Again, using this comparative genomics approach to identify these human-specific elements. I will give you one example of a novel, a quote-unquote novel human-specific element that we identified as being perhaps important for human cognition, the gene clock. And then I'll give you an example of how we're using human brain activity to identify genes involved in human cognition. So a lot of our work is predicated on this hypothesis that vulnerability to disorders of cognition emerged as a consequence of the evolution of cognition. And so by studying evolution, this may give us some insight into cognitive disorders and vice versa. Now, when we look at this image of a human brain next to that of our closest genetic relative, a chimpanzee, it's clear that there's a big difference in brain size, but clearly brain size is not sufficient to distinguish the vast phenotypic differences between humans and our closest genetic relatives. And there's been a lot of work on many levels to study both similarities and differences in the brains of humans and non-human primates. But a lot of our work was inspired by the work of King and Wilson many years ago that were looking at protein sequences between humans and chimpanzees and found that they were very similar and came to the conclusion that regulatory mutations may account for their biological differences. And with the subsequent sequencing of the human and chimpanzee genome decades later, that was really borne out, showing that encoding regions very similar, but there's a lot of differences that are lying in these regulatory elements. And so that has inspired a lot of the work that I will show you here today. And so to test this hypothesis of looking at gene regulation being perhaps changing on the human lineage, a number of years ago we carried out what we thought of as a pilot project comparing three brain regions, frontal pole, hippocampus, and caudate nucleus, in three species, human, chimpanzee, and rhesus macaque. And the reason we chose these three particular brain regions was that we could be very certain that we were really comparing apples to apples, so the same brain regions. And so we wanted to identify things that were changing specifically on the human lineage. So if one is only comparing human and chimpanzee, one cannot assign whether that change is changing either on the human or the chimpanzee lineage. And so in this case, we're including an outgroup, the rhesus macaque, for comparison. And in that case, we can assign in which direction genes are changing, so things that are the same between chimpanzee and rhesus macaque, but then are different on the human lineage. And so this is just showing, based on the fossil record, that this recapitulates this phylogeny, right, that rhesus macaque is our outgroup. And now if we start to do some gene expression analysis from the brain tissue of these three primates and look at genes that are going up or down, you basically recapitulate the same sort of phylogenetic tree. However, what I'm going to show you in the next few slides is that if we start to think about these large gene expression data sets in a different manner, and that there's some other underlying structure, namely how large groups of genes are co-expressed, we can start to identify additional information. And so this is just a few slides kind of schematizing what I mean. So again, here's our idealized differential gene expression analysis. You have genes going up in control and down in experimental. In this case, each row indicates a specific gene, and so you could say a specific gene is going up or down in one versus another. However, it becomes very difficult to then figure out how to prioritize genes. So each gene is treated as an individual entity, and we really don't know how to move forward. And so you can say, well, I like this gene because it has the smallest p-value, or I like this gene because my neighbor lab down the hall has the knockout mouse for this gene. And those are all valid approaches for functional follow-up. But I'd like to show you how you may be missing a lot of the data by taking that approach. And so again, just simplifying the data in this manner, if one looks at gene 1, and this is expression in control versus experimental, you can see that the expression of this gene is very similar between the two groups, non-significant. The same would be for gene 2, between control and experimental, not very different, non-significant. However, if you now take the same data and kind of turn it on its side and look at how these two genes are changing relative to one another, so here's gene 1 and 2 across all of the samples, what you can see is now in the control group that there's a highly significant correlation between these two genes across the control samples, and a non-significant correlation with the experimental. So taking this co-expression approach, now these two genes become important. But if one were taking only a differential expression approach, you would have not examined, perhaps, these particular genes. And so what we do is we take this co-expression approach across all of the genes and all of the samples, and then we observe how they are related to one another and put them into modules and identify hub genes. And a pretty nice way of thinking about a hub gene, I like to always show this example, is a visualization of the American Airlines map. Because in this case, Dallas-Fort Worth, where I'm coming from, looks very important. It's a hub. And so when we have our unusual snow, ice once a year in January, like we had in the last 48 hours, if you're flying American Airlines anywhere around the world, you may have some travel delays because of Dallas-Fort Worth being very important. So the same is true for the gene co-expression network approach. If one can identify hub genes, what we and others have shown over a number of years is that those genes that are hub genes really drive the connectivity, drive the co-expression. And if you did the same sort of network, taking, say, a knockout of that particular gene, the whole co-expression may fall apart. And so we applied this approach to our comparative genomics, and what we found is that by looking at gene connectivity, now we saw accelerated evolution on the human lineage. So this was something that we could really only pull out of the data by looking at how large groups of genes are co-regulated, and this was something that was, again, predicted many years ago by King and Wilson. So what we did next was put all of these modules of co-expressed genes and assign them to species indicated here in color and brain region. There are lots of modules we didn't really explore in terms of what could be driving them. But for my purposes, I was really interested in what was important for the human frontal pole specific networks, because we know the frontal pole has been modified in the human lineage and that it's frequently disrupted or at risk in many cognitive disorders such as autism and schizophrenia. And so I wanted to dig down deeper into these human specific frontal pole co-expression modules. And one that really struck me as being exciting and interesting to explore was this one particular module that had the gene clock as its most highly connected hub. So clock has been identified as a gene involved in circadian rhythms, and it hasn't really to date been studied for its role in cognition or its role in the cortex in general. And what was also striking was when we looked at what the other genes were that were co-expressed with clock in this human frontal pole network, none of them were the kind of canonical circadian rhythm genes or known targets of clock in other tissues. But what we found was that, in fact, this module was enriched for genes involved in cognitive disorders rather than these circadian genes. And so I thought this would be a good example of something that I could follow up functionally and really explore in the lab. And now one caveat to keep in mind was that all of this comparative genomics data was taken from post-mortem brain tissue. And so one of the questions that I frequently received when I first was presenting these data was, well, how do you know that this isn't really driven by time of death of the samples from which that you were examining? And so I was really pleased when this paper came out right after ours from Huda Kill and William Bunny, where they had taken post-mortem, human post-mortem cortical samples where they knew time of death, and they could plot the expression of genes over a circadian time. And what they had found was that many of the canonical circadian factors, such as Arntal, PER1, PER2, et cetera, did have this cyclical expression, but clock did not. So this fit with our thinking that perhaps there was something unique about clock increased connectivity, and in fact, it also has increased expression in the human frontal pole. And so this led to a hypothesis that increased clock expression in the human brain may have been adapted for a non-circadian role related to cognition. And to test this hypothesis, we took a number of approaches. So one was, again, to use human post-mortem tissue that we could examine clock targets by doing chip sequencing. Another was to use a model of human fetal brain development that we and others use, where we can culture these human fetal progenitors in the lab and also turn them into post-mitotic neurons. And much of the gene expression in these post-mitotic human neurons recapitulates what's found in vivo in human fetal brain. And so a talented MD PhD student in the lab, Miles, who graduated last year, undertook this project. And he carried out chip sequencing from frontal pole, as well as another area of the brain that we had shown had increased connectivity for clock in human compared to non-human primate brain. He found that these brain regions had some overlapping regions of the genome bound by clock, but they also had some distinct bound regions. In addition, he found that these bound regions were enriched for E-box motif, which clock is known to bind to, so that was a nice positive result. In addition, many of the bound regions are found in promoters of genes, again, fitting with this idea of clock being a transcription factor, and it's also a known transcriptional activator. So this has now identified a number of potential in vivo direct targets of clock in the human brain. But we wanted to use a more genetically manipulable system so we could test causality of clock expression in human neurons. So we turned to this model of the human fetal progenitors that we can differentiate into neurons. So Miles differentiated these cells for 4 weeks into neurons. He then transduced them with a lentivirus to knock down expression of clock, or a control. He synchronized the cells because they do have an inherent cyclical rhythm. And then every 4 hours over 2 days, he extracted RNA and did RNA sequencing to look at gene expression. And what I hope you can see is very clear here. So this is genotype knockdown cells versus control. We were able to identify many differentially expressed genes with clock knockdown. However, we didn't see an effect of gene expression over time. And so this fit with our hypothesis that perhaps clock could affect gene expression independent of time. In addition, we carried out this co-expression network approach to try and hone in on what might be some of the more salient features of what clock might be regulating in human neurons. And one module that struck us was this one because it was enriched for genes involved in neuronal migration or cell migration. We thought, well, this is great. This is something that we could actually go into the lab and test a hypothesis. And so what Miles did is he grew a neurosphere. And so we took these neural progenitors, grew them as balls of cells, and then he co-transduced the cells with a hairpin to clock or control, and then measured how far they migrated out from the center. So this doesn't recapitulate in vivo brain neuronal migration, but at least it's a test of, did the genes that we identified from the RNA-seq, can we really prove that this is functionally relevant based on the gene ontology? And so what Miles found was that with the knockdown of clock, he saw an increase in neuronal migration. And this was really exciting for us, that we could actually use this cellular system to prove what we had identified through the transcriptomics. And this presents the possibility that perhaps clock is involved in either inhibiting migration in the developing brain or is in some manner promoting proliferation or keeping cells in a non-migrating state, perhaps in this outer ventricular zone that's been shown to be expanded in the human brain. And so this is something that we can now, and we are testing in human brain organoids that was already introduced to us earlier in this symposium. And so we're excited to be moving in this direction. In addition to following up on this in organoids, we are also following up in a number of other manners. This is one other module from his data set that I wanted to show to you because it ties back into the evolution concept of clock. And so in this particular module, what we had found was that there was an enrichment of genes involved in autism and intellectual disability, as well as genes that have been shown to have human-specific expression, taken from my previous work, as well as a number of other studies in the literature. And what Miles then tested was whether the hub genes in this particular module were different than the hub genes in the entire network. And what he found was that the human-specific genes tended to have higher connectivity or tended to be more hub-like in this particular module than in the other modules. So this suggested we could really use this network approach to hone in on what might be the most important genes that are changing in expression with manipulation of clock in human neurons and relate this back to this evolutionary question. And so what this shows, or what I hope I've shown you, is that comparative genomics hypothesized clock as having human-specific non-circadian function. And now we have data to support this through transcriptional regulation of pathways important for neuronal migration, cognitive disorders, and human-evolved gene networks. And so what we're now doing, in addition to the organoids to follow up on this, is using a humanized mouse model. And so what we've done is we've made a back transgenic where we've taken the human gene, all of the regulatory, well, at least flanking regulatory regions of clock, put it into a mouse, and then cross that to a clock knockout mouse. And so what we can now test is how transcriptional networks might change, how behavior might change. Of course, we're only changing one gene, but there is precedence in the literature that manipulation of single genes can lead to alterations in behavior of animal model systems. But even while I think these mouse models and other model systems are important, at the end of the day, the mouse is still not a small human. And so while we're using these approaches in the lab, I'm still interested in how we can use more data from humans to really explore this idea of human-evolved cognition. So what can we do? And in particular, how can we really get at the molecular mechanisms of human cognition, which I think is a challenging question, and why we're all here, genetic and molecular mechanisms. So of course we can do behavioral studies, imaging studies we've already heard about, and we can take the peripheral tissue, we can use those to make the organoids, as was already mentioned, and we can profile post-mortem brain tissue. But I wanted, when I had first started thinking about this, wanted to ask how we can sort of integrate these across systems and across different diseases. And I was really inspired by a lot of the work that had been done with GWAS studies in brain imaging, structure and function. And I thought, well, why can't we take that one step further and go one level up from DNA to RNA to look at gene expression and how that might correlate with functional readouts from imaging such as fMRI. And so this was published a number of years ago, and this was work from a talented postdoc in the lab who now has his own lab in China. And we asked the question, could we correlate brain gene expression with brain activity as measured by resting state fMRI? And so I'd like to emphasize that these data are they are derived from two independent populations. So this is post-mortem brain gene expression, because that's one of the only ways in which we can obtain RNA from human brains. And these are resting state fMRI from different people. And so when we started this project, I thought it was a bit risky for my postdoc to take on, because I assumed, well, it's an interesting question, we can do it, but we're probably not going to find much. But he was game, and so we used a number of data sets. We had our own RNA-seq data set taken from throughout the human neocortex and two publicly available data sets. We also used publicly available resting state fMRI from the Functional Connectomes project, and we were pretty careful in that we did these correlations across all 5 data sets, and we had replication, and things had to be consistently correlated. And to my surprise was we were indeed able to identify over 30 genes that were either positively or negatively correlated with the resting state fMRI and the default mode network with this post-mortem brain gene expression. And a lot of these genes were very interesting. They were genes involved in ion channel function. And so in addition to us identifying these genes, I was very happy when a number of papers came out, including work from here at the Allen Institute, showing a similar ability to find correlations between postmortem brain gene expression and fMRI. Different approaches, different data sets, yet we're all kind of demonstrating the same sort of thing. And I think this opens up the possibility of doing these sorts of approaches using all sorts of publicly available or data sets we're generating from different diseases, such as was mentioned previously, from autism or from schizophrenia or any other trait or disease of interest. But this was all from resting state fMRI, and what I wanted to next ask was, could we do the same sort of thing doing some sort of, while the individuals were doing a functional task? And if we did this same approach and identified the same genes, then I'd start to worry about how we were doing this analysis or how we were interpreting it. And so I was really lucky that a colleague of mine at UT Southwestern, Brad Lega, who's a neurosurgeon, was collecting data from human patients. So these are patients with epilepsy, and they're unable to treat their epilepsy through medication. And so they're undergoing some invasive queries to identify the site, and then tissue is removed. But Lega is very interested in memory, and so while these patients are undergoing intracranial EEG to record where their epilepsy is occurring, he's carrying out a memory task. So the patients are shown a series of words. They're then given a distractor task where they're asked to do some math, and then they're asked to recall the words that they were seeing. And throughout the entire period, they're recording intracranial readouts through various frequency. And one can assign what they call a subsequent memory effect to a positive or a negative recall during this task. And so we took those data, and we again compared those data to the post-mortem brain gene expression from throughout the neocortex. So it was basically the same data set that we had correlated with the resting state fMRI. And we were able to break down correlations based on the various wavelengths that they're recording during the intracranial EEG. And to our surprise, what we found was that the majority, first of all, that we could identify genes that were correlated with this SME paradigm, and that the majority of these genes correlated with beta oscillations. At first, this was a bit surprising because the literature had really pointed to gamma oscillations being involved in this memory paradigm. But a lot of those studies in both human and rodent models had been using the hippocampus for their approach. And so there was some precedence for beta oscillations in the cortex being important for this effect. In addition, if we looked at the gene ontology of the genes that we had found correlated with SMEs, again, it kind of fit in terms of neuronal function and genes that had previously been implicated primarily through patients with various diseases or animal model, genes involved in memory or cognition. Many of these were the same genes that we identified as being correlated with SMEs. In addition, we could take these data and see how they fit with these large GWAS that were available. In particular, many of the cognitive GWAS, such as ADHD, bipolar, major depression, and autism. And what we found is that there was significant enrichment for these SME-correlated genes in the cognitive disorder GWAS. Schizophrenia was the only one that didn't fit this. And then the non-brain-related GWAS, such as these, were not correlated. So this was a really nice confirmation that perhaps what we were identifying really fit with what was there in the literature. In addition, we took this co-expression network approach to, again, try and prioritize which genes might be the important ones for further functional study. We had identified almost 200 genes that were correlated with these SMEs. So if someone in my lab was going to go off and start their own lab and work on one of these so-called new human memory genes, which one would they want to focus on? And so again, taking this network approach, we were able to identify 2 modules that were enriched for the SME-correlated genes. And both of these were very interesting modules, and I'm just going to highlight one of them here for you today. And this was pretty interesting to us, and this was enriched for genes involved in synaptic transmission and cation channel activity. And one of the hub genes in this module, this GRM2, is a metabotropic glutamate receptor, and a number of other metabotropic glutamate receptors have been implicated in other cognitive disorders such as autism. So this is really exciting to us that perhaps this, if one were to do some functional follow-up, might be a salient gene to further study. In addition, this module was enriched for a number of disease-related genes in general, such as schizophrenia and autism. And so we really think that this co-expression approach, again, applied to these various data sets, in this case, intracranial EEG, memory task-related genes, could provide some novel insights into which genes are important for further functional study. Now, I mentioned that one of the impetuses for this was to really see if the genes we had identified from correlations with resting state fMRI would be the same or different than the ones that we would correlate with some sort of functional task. And to our delight, the SME-correlated genes were mostly distinct from those that had been correlated with at least 2 independent studies of resting state fMRI. In addition, since we didn't really have a replication cohort for this study, it was from one large SME data set, we wanted to make sure that the genes we were identifying weren't just false positives from some aspect of the way that we were doing the calling of the SMEs or the correlations. And so what we were able to take from the same patients was the information, the intracranial EEG recordings during the math distractor task. Run the same correlation, see if we get the same or different genes. And what we found was that we could identify genes correlated with this math task. They correlated mostly with low gamma oscillations, so this was different than the beta oscillations we had identified with SMEs. And in addition, these genes, again, were mostly distinct from either the genes correlated with the SMEs, and however, there was some overlap with the resting state fMRI. So again, there's some interesting genes here at the intersection of these different approaches that might be worthwhile exploring in functional studies. And so just to conclude, hopefully I've convinced you that a comparative genomics approach, and in particular a co-expression network approach as opposed to a differential expression approach, really can help us in uncovering genes that are important and that are changing uniquely in the human brain. Through this approach, we were able to identify one transcription factor clock that may have been recruited to have differential transcriptional targets in human neurons outside of its role in circadian rhythms. And that by taking this population comparison of human brain gene expression and activity, we can now start to identify genes involved in cognition. So just to emphasize again, this was a population approach of post-mortem brain gene expression and either resting state fMRI or intracranial EEG. And while this has proven to, I think, give us some useful information, what we're now doing, and I'm really excited, is some within subject study. So we have individuals for which they're undergoing intracranial EEG. They're also undergoing a number of imaging studies with the same sort of memory tasks. And we're obtaining brain tissue from these individuals because frequently the neurosurgeon needs to remove unaffected tissue to get to the site of epilepsy. And so we can compare unaffected brain gene expression to measurements of memory, measurements of brain activity, in the same individuals. And so this is really exciting for me to move beyond even doing these population comparisons. So I think I showed most of the people involved in the work. I'd like to also mention another postdoc in the lab, Stefano, who's involved in the intracranial EEG study, collaborators Dan Geschwin and Todd Preuss, who collaborated on the comparative genomics approach, Joe Takahashi and Carol Tominga, who collaborated on the clock study, and for my imaging collaborator, Hanjan Liu, and then again, Brad Lega for the intracranial EEG. My funding sources. Thank you for your attention. I'm happy to take questions.
Okay, we can take questions now.
I'm curious with clock, the gene. What you presented is genomic comparison, comparative genomic. If you are looking on GWAS, do we have evidence that clock is associated with some of these traits? And if not, why we are not able to reconciliate comparative genomics and studying population polymorphism in human, for human brain in particular?
Sure. So there's at least one study showing association of clock with sleep, I think it was. And, but from the disease kind of GWAS, it hasn't popped out yet. I mean, I think that those are still, I'm going to go out on a limb in this crowd, maybe underpowered. So there's also been some work in lymphoblasts from patients with autism showing clock is differentially regulated in cases versus control. So there's some minimal evidence, I think, that it may be important, but I think we're still at early stages. Why? And then I think it was really a two-part question, right? Why do the comparative genomics and the GWAS? Why or why not are we seeing more integration? Well, it's interesting from some of the earlier talks today, as I was seeing genes up on the screen, a lot of them are ones that we've pulled out either from functional studies or comparative genomics. So I think there is some integration, and maybe those are some future avenues of research of integrating all of those data sets. But I would say the comparative genomics has also been underpowered. We've really been limited by the availability of non-human primate brain tissue samples. And so our study and most of the other studies to date, there was a recent one from Nanad Seston in Science. I mean, we have an N of 5 usually for chimpanzee, for example. And so I think, if possible, more could be done there as well. And then maybe we would start to see them come together.
Question in the middle right there.
I was wondering, in your brain and genetic data, for example, fMRI or ECOG data, you look at how the gene expression correlated with fMRI signal or ECOG signal. So as far as we know, the ECOG sample is usually pretty small. So I wonder that how do you choose what genes to explore? It depending on the region that they recorded a signal. Like for instance, you say that the theta oscillation or the gamma oscillation. So it depends on the region that show the oscillation that was correlated with the behavior, and you look at the post-mortem gene expression in that region and you do the analysis. Or I just want, can you clarify what type of approach you use to choose a gene, and how do they survive the correction for multiple comparisons? Yeah.
Right. So in our case, for the ECOG, intracranial EEG data, we actually had pulled from all of the available data. So it was actually almost a couple hundred individuals, which was on par with actually what we had used for our resting state fMRI. But our correlations were based on how the expression correlated across the brain region. So we actually didn't, so one would not assign that the correlation was specific to a particular brain region. It's kind of like the network approach, where it's how the expression of the gene varies across the regions, as well as the oscillations during the memory recall task. So hope that maybe wasn't quite clear. But what I am thinking is the way to do the functional follow-up is to take the network approach, where we're taking the co-expression and then we're integrating that with the genes we identified, correlating with the ECOG data, and trying to determine which ones are the ones that are really driving the co-expression, or are there any really driving the co-expression. And so the modules that are enriched for these SME-correlated genes are in my mind, most likely to be the ones involved in the behavior that we're using to make the correlation, in this case, the SME, the memory paradigm. And so that's how we're trying to identify which genes to follow up, by taking the co-expression network approach and integrating it with the correlated genes.
Okay, thank you very much.